Okay, so maybe we should start with a little explanation. She's got a little.
Dear. we,
There must be a good story in it.
no, there is not a good story. I slipped upstairs in the hall in front of my office and slid and rammed my hand into Arno Metulski's, maybe this is a good story, Arno Metulski's file cabinet, which is in the hall. And it's very, very heavy. So I stopped abruptly.
This is the first time in 20 years I've seen Carol with any injury like this.
I think I've never broken a bone before. So anyway, let's move on to something else.
So the first thing I'd like to ask you about is you arrived here at the University of Washington in 1976. Can you tell us a little bit about how you got here to the University of Washington and the Department of Genetics?
Well, we drove, actually. And we came from California, so it was a rather long trip. How did I really get here? I got here because of an amazing person at Caltech, a guy that some of you probably know, and turned out to have been a friend of Herschel's, which is not incidental, I'm sure. Ray Owen, who was a very special person at Caltech. And there was an ad in Science that was was I think for an immunogeneticist, which I was only sort of. And I came into Ray's office and said, "Hey, there's this really interesting advertisement for University of Washington, Department of Genetics, etc." And he said, "Oh, really? I know someone there." And he went back to his phone, called Herschel, and put me on the phone out of the blue. I have no recollection of what I said, if anything, and that started the whole process. I did apply, and then it worked.
Well, what was special about coming up to Seattle and the University of Washington?
What was most special? I mean, this is someplace that Tom and I really had admired. We were at Friday Harbor. Tom had a NSF fellowship in 1970 at the Friday Harbor Lab. And so we had really a good idea that it would be wonderful to live here. And when this opportunity came up, we were happy to do it. So we did.
And when you arrived here, can you tell us a little bit about what type of experiments and what sort of research directions you were doing?
I did do immunology in a way. It was really more molecular biology of cells of the immune system. So I taught immunogenetics to graduate students and developmental genetics related to that kind of thing. So I learned a fair bit of immunogenetics in the course of that. But we were really doing molecular biology and control of transcription in B lymphocytes.
And how did you get interested in that area?
As a result of my graduate work, in my graduate work I was studying T lymphocytes, the other kind, and using a sort of a genetic approach to try to study steroid hormone action. Turns out glucocorticoid hormones kill immature T lymphocytes. And so you could do some selections, you could do a bunch of things with those lymphocytes. And toward the end, when I was writing my thesis, I thought, you know, I should probably learn something about what these cells really do, which I really hadn't done up to that point, because nobody else in the lab worked on them. And I got really interested, and we decided, I decided that that might be something interesting to go into. So I did a postdoc then on that, sort of on immunology.
So the other question I had is, when you got up here to Seattle, you had a department with Drosophila people and yeast people. What were some of the special things of the group up here that helped you with your immunogenetics research?
The two people who were most helpful were Stan Gartler and Don Pius, who was a joint, I don't know if he had a joint appointment adjunct. He was in pediatrics, but he did things that were really very similar to what I was trying to do. So those two people, Stan, who worked on mammalian cells, and and Don, who worked on B lymphocytes in another context. So they were both really crucial people, helping to get me started in the research end.
And what about the interaction with your peers?
I mean, actually, the other person who was really crucial to me was Larry Sandler. Larry was just down the hall from me and had kind of taken me under his wing and helped me get started, gave me a lot of good advice about how I should try to act as a grown-up and things like that, which was useful. and actually, he then was really instrumental when I began to teach. I listened to all of Larry's lectures, and at the end of it, unfortunately, all I'd learned is that I couldn't do that. And so then I had to come up with a different approach that might have worked. I don't know that I ever did, but anyway, his style was so perfect for him and so imperfect for me that I didn't even try to adopt it. But he was a huge help to me. I still have a chair in my office that you've sat in, actually, that Larry gave me. So.
Well, the next thing I'm curious about, here you are up here in Seattle, you're doing immunologically oriented research. Mm-hmm. At some point you made a departure for that. What brought that about?
I'm not really sure. I think I don't have an awfully long attention span. And so, after some length of time, I began to get restless. I wanted to do something different. And this department was unique in giving me the opportunity to do that. Well, as you know, you were part of that whole transition from B lymphocytes to malaria, which is not an obvious switch, to say the least. But several people were really crucial in that. Well, two people in particular. Lee Hartwell, who worked with us on the idea, as you probably remember, and then Breck Byers, who was chair at the time, who supported. I mean, you really don't want your faculty to suddenly do something completely and utterly different generally. It's just not the usual approach. And Breck and Lee both supported, as you remember, supported us as we began to use yeast to study malaria, and supported Tori Brophy, one of the students, longer than they normally would have done, et cetera, et cetera. So it was a transition that took a couple of years and wouldn't have happened, I think, in most other departments.
So I guess one of the things I'm curious about, and I never asked you about this, there are a lot of different directions you could have gone, but you headed into tropical diseases and malaria.
Mm-hmm. What do you want to know about it?
Why did you go that direction? What attracted you to the issues of malaria?
I'm not exactly sure. It started with a journal club, like I think a lot of things in the department as well. And I chose a journal club that had to do with malaria. And as I was doing the background research, just to make sure I knew at least a little bit when I gave the journal club, a lot of things came up, including they kept saying, drug resistance is a really big deal and nobody really knows how to attack it, nobody knows how to work on it. I thought, well, gosh, I could think of what to do about that, perhaps. And once I got that idea, and then we began to sort of work on it and finally got the idea to express the malaria genes in yeast, things really began to happen. So I don't really know why I chose that particularly. It did appeal to me, obviously, quickly. I decided that it was something. I like doing things that nobody else does. Maybe you remember, but we were studying a B lymphocyte cell in which the now very famous factor NF-kappa B had been discovered. And people began to work on it, and it turns out it's everywhere. It does everything in all cells, as far as I can tell, including Drosophila. And it just came to the point where you sort of ask yourself, well, if we don't do this experiment, will somebody else do it? And when the answer to that is yes, you think, well, gosh, maybe I should go off and do something where it isn't quite so obvious what should be done. And so that was very attractive because there was almost nobody working on malaria in that context at the time.
So I guess the other thing I'm curious about when you got into this malaria project here in this environment, what were some of the special things about here at the UW that allow you to do something as crazy as taking proteins and genes from malaria and putting them into yeast cells?
Well, I mean, as you know, Lee's lab was already deeply involved in expressing human genes in yeast, and he had started on his now very productive line of reasoning that you could actually study, in that case, cell cycle and cell cycle genes in humans. And I thought, well, gosh, you know, maybe we could do that take malaria proteins and do something similar. And you know, for most malaria proteins, it in fact would not have worked. But the one we chose had a lot of special properties, and it worked very well. It took quite a long time. I mean, the malaria field was not notably enthusiastic about our studying malaria in yeast. I mean, we got lots of negative feedback for a while on that, but ultimately it worked out well, and so we got a huge amount of mileage out of it, in fact.
And basically at this point, that system you came up in yeast has been described by some as studying Africa in a test tube.
Yes, by our favorite person, yes.
And so I guess how has this work contributed to the problems of drug resistance?
And mostly it gave a very straightforward way of studying one class of antimalarial drugs, and because of the awesome power of yeast genetics, in fact, we could begin to even do things like taking a finger stick sample from a person who has malaria, PCR amplifying it, and then co-transforming that with the appropriate vector into yeast, and then you simply select with the antimalarial drug of interest, and you can find really rare alleles of the appropriate gene that's in the field. And it took us quite a while to sort of realize that this was going to be possible. But that was something that really can't be done by just looking at the malaria genome itself. It could be now, actually, but at the time there really wasn't any way to find extremely rare alleles that were in the field, sort of sitting there waiting for the drug selection to take over. So that was probably the most significant of all the various things that we did, I would say, in terms of impact on the field.
So you started out originally asking questions about drug resistance to certain drugs, but now there's, in addition to that, other unexpected applications of this work.
Well, yeah. I mean, other people have used it in a whole variety of ways in malaria, and in fact in other pathogens as well. Again, this is hardly our idea. Once you understand that yeast can express proteins of all kinds with the appropriate promoters and so on, a huge amount has been done by many, many people in many fields expressing other kinds of genes because of the ease of using yeast. It's become a very almost standard sort of approach, long before we did it, in fact. One of the other things I'm curious about, what are some of the changes in science with the technology that has enabled you to do this type of project? Well, obviously there have been huge changes. I mean, well, as I just described it, PCR was crucial to the ability to do that. And PCR is straightforward enough. It's the kind of thing that can be done even in lots and lots of our African colleagues' labs. There's a PCR machine and sufficient training so that people can do lots of things. Without that certainly wouldn't have happened. I mean, at this point now, the other nice thing is the malaria genome is about the same size as the yeast genome, And so, you know, this is a few hours on your favorite aluminum machine or whatever. And that's allowed some of my, we have a very close collaboration in the current project with the Sanger Center. And they've come to the point where they've worked out a lot of their bioinformatics on the malaria genome simply because it's so ridiculously small from their point of view. And they then develop the bioinformatics, the various algorithms they need, and apply those then to the much larger human or whatever other genome they're working on. And so it's sort of interesting to have seen malaria become, in that sort of strange sense, a kind of a model organism for humans, which is certainly not, in terms of the biology, not what one would have expected. Excuse me, would have expected at all.
So your work has a very strong connection to the field.
It does now, yes.
Can you share with us some of the things that's fun about that?
Yeah, I think the most fun about it is the people who work there. Working in the field requires a certain sense of humor and sort of idiosyncratic approach to life. And so the people who do parasitology, especially those who are doing the field and doing clinical work, are just an extremely interesting group of people, way more interesting than immunologists ever were. I mean, and the other thing I really have always liked, if you go to a meeting, if you go to, again, an immunology meeting, you see Caucasian faces and East Asian faces. Lots of Japanese and Chinese scientists are involved in immunology. But that's about it. If you go to an international meeting on parasitology, especially on malaria, you see the world. And I really like that. You know, I have friends all over the world, and via Skype, I can even talk to them a good fraction of the time. And so that's been a really wonderful way, as I come toward the end of my career, to see how science can move from the, you know, very detailed bench kind of things that we were doing for a long time into this much wider vista and you can really see the impact of what you're doing, and that's also really satisfying.
Or can you tell us a little bit about some of the fascinating people in your group who have helped with this project, or who the project succeeded in spite of them? And I'm excluded from any comments you're about to say.
This is what's called a leading question, right? So, no. So Jason is well. Some of you, all of you, are laughing. Know this, but when I was trying to make this transition from immunology to malaria, Jason was just beginning his graduate career, and he really insisted that he was going to do this, and I really tried to talk him out of it. I mean, it's a terrible plan to go with an advisor who is doing something completely new that she knows nothing about, that may or may not work in the time scale of, you know, geologic time. And Jason insisted that he wanted to do this, and he was going to do this, and he did in the end do it. The other person who was really impressive is Tori Hurtle, now Tori Brophy, who was working on her project, which was part of the immunology part, and about halfway through, after a couple of years, she'd finished what she was doing on that particular phase, published a paper or two on it, and you know, I said, look, Tori, I am committed to your being able to finish what you started. There's no reason for you to change. And at that point, she said, you know, I really think this is pretty interesting. I think I'm going to change to do this. And again, I tried to talk her out of it. But in fact, in 2 years, she worked on something very similar on a different pathogen called Cryptosporidium, and published, again, several papers on that. And finally, her thesis then had the crypto work that she had done, and her immunology work was just a couple of papers that were in the, sort of an appendix at the end. And I thought, boy, this is amazing, that when you have students like that, it's pretty hard to fail, I think, because these were both extraordinary things, both for Jason to to do this in the first place, and for Tori to make that change at that point in her graduate career. I mean, she graduated in under 5 years, I think. So those were both pretty amazing, and I think probably wouldn't have happened in most places. So I was lucky.
It must have been something in the water in J-Wing.
Yeah, right. Could be. It could be.
So you started out doing this interesting project as a geneticist to help with malaria in the lab. It's now taken a different direction, your work where you're doing things outside of the lab. Can you tell us a little bit about that?
Yeah, I mean, it was a sort of a natural evolution. As I said a couple of minutes ago, once we figured out that you could actually get information about what's going on in the field, in the genetics of the parasites, by PCR copies and putting those into the appropriate vector and into the yeast and selecting, then people got really interested and said, oh, well, why don't you look at our samples? And, oh, we have samples from here. Oh, I wonder what's going on over there. And we ended up developing quite an extensive network of people, sort of doing that as a sort of as a service almost. So we would, I'm not sure it's legal anymore, but we were getting, you get them in the mail. Blood spots it turns out the DNA is very stable, and as long as it's kept dry, all they have to do is, the child comes with malaria, they do a finger stick, which they have to do anyway to diagnose whether they have malaria or not. And you can essentially blot it onto the filter paper, dry it, and mail it off. And so people began to do a great deal of that. And I got more and more interested in the sort of epidemiology of what was going on, and had more and more resources in the sense of a network of people who had similar interests, and they had the samples. And so we did a great deal of that over a period of several years. And the more we did, the more clear it became that this was something that was really worth doing and that nobody else was doing. And you'd go to a meeting and some bright young person would get up and tell you what was going on in terms of the resistance of the parasites in their place. Today this is what I found, and then they'd sit down and somebody else would get up from another place and say, this is what's going on in drug resistance in the parasites in my place now. And this was important information, but you could not put it together. Students would later students, people like Laura Certain and Vivian Hawkins, were doing lab experiments, and when they'd come to the end and need to write the introduction to their thesis, or hopefully a bit earlier than that, they'd complain that there was a lot of information about what was going on in terms of drug resistance across the world, but there was no one place where you could find it. And that's in fact what's really needed. I mean, if you're the Minister of Health of Nigeria, you really would not only like to, but need to know what's going on in terms of drug resistance all over your country, and in fact all the countries that border yours. And so we ended up developing a network to collect this information, to put it in a database that would be user-friendly and would allow lots of bells, many bells and whistles, but allow then these kinds of information to be pooled in an appropriate, statistically appropriate way, so that you can then make maps and begin to do models, look at how drug resistance against one drug moved from this place to that place to this place over what time, and use that to try to predict what will go on with resistance to another drug. I mean, the current Southeast Asia, unfortunately, is a sort of a source of most of the drug resistance. And currently the last drug, called artemisinin, is now beginning to be less effective in Cambodia and in the rest of the Mekong in fact. And so you would really like to know what have been the patterns of change over both the time period and the geography of what happened in terms of drug resistance for other drugs And to see, it's not clear that that would be exactly the same, obviously, for another drug. The selective patterns are quite different and so on. Nonetheless, this kind of information, once you've got that kind of information, you can begin to use models and try to make some predictions about what might happen and where containment of the malaria would be most effective and things of that kind. So it has a very practical sort of orientation at this point. And it's really fun, I must say. I mean, this is really people all over the world. So I think we keep thinking we should somehow thank Skype, which has made this project possible. Now that it's owned by Microsoft, I don't know that we need to thank them, but anyway, you should probably edit that out, actually. The project is funded by the Gates Foundation, so all of that can go away. Anyway. Yeah.
So this area of this question of drug resistance and malaria, that's one area where you were able to contribute here in Seattle as a geneticist. Mm-hmm. There's another area that I think because of the uniqueness of this department you were able to contribute that had to do about all the different strains and tracking the strains. Can you share a little bit about that?
Right. When we first began to try to study malaria, the first thing we did was to get a bunch of different malaria strains and begin to grow them in the lab. And the first question was, since we were geneticists, Jason and I, well, we needed to have some markers so that we could tell them apart. And it turned out that the history of none of these strains was known. It was really the equivalent of, well, I got it from George, and I think he got it from, you know, Joanne, and no, I don't really know where this one is from. And even worse, that nobody seemed to bother to keep track of them in the lab. It was really quite astounding. And so malaria have a number of genes which are really unusual, whose function is really not understood. But what's unique about them is that they have very different repeats in the middle of them, and then the flanks are the same. And so Jason devised a system of the sort of obvious to us, you know, primers on both sides in the conserved area, and then different strains have different size repeats. Very straightforward. And actually did that, showed that you could tell the difference, et cetera, and published it. And people kept coming up and saying, wow, that was a fantastic idea. And you think, really? I mean, you know, it's pretty straightforward. But it was the first foray into any sort of genetics. There was almost no genetics of malaria. That isn't quite true. There was one cross done at NIH by Tom Wellams a long time ago, which has now been used over and over and over again very productively. But it's so expensive. I mean, you need a primate intermediary, you need a huge amount of money, you need a very long time scale, and Tom has been at NIH for, I don't know, 30-some years. But that's the only cross, that, you know, the only real genetics that's been done on human malaria in the whole time. But yeah, so genetics has not been a huge thing. I mean, genomics is now a huge deal, as I said, but straightforward, you know, transmission genetics has not been a major part.
And you guys would be happy to know we did not do any crosses in the department back in the 90s.
Well, when we started to do this, there were many questions. I mean, you know, my colleagues were more than supportive, as I have said, not just about this, but about everything. And but there were a lot of questions when we said we were going to grow malaria parasites in the lab, and you grow them then in human red cells. You need a hood, which we already had because we'd worked on mammalian cells and all. And people would sort of sidle up to the question, but the gist of it was, can I get malaria if you're going to grow it in the lab? And the straight answer is, no, you need a mosquito. and so there's no, in that sense, I mean, there's some danger from human blood and, you know, hepatitis and all kinds of things like that, but there's no danger of getting malaria. Even if you stuck yourself it would be pretty tough to give yourself enough of an infusion to really come down with a case. You probably could. If you had a full transfusion, you certainly could, but beyond that, there's no danger. But nobody said we shouldn't do it. It was just kind of like, should I avoid your lab from now on? You know, I won't come to that corner of the J-wing, or something like that. Yeah.
So, which leads me to the other question. You talked a little bit about genomics. What is, over the years, what are some of the changes that you've seen in genetics that has evolved the project?
Right. I mean, none of these are things I've done, but obviously as genomics has advanced so much based on the kinds of things that you all do, in fact. One really nice thing, obviously nobody else in the department, except for Ray maybe, works a little bit on malaria, but that's not a major thing for most of the people in the department. But there's been a huge advantage for sitting through all of these journal clubs and research reports. I mean, obviously the first 10 years when we heard about the yeast cell cycle, you know, multiple times a week, you know, and after a while you think, you know, okay, I've seen that before. But when we began to actually work with yeast, you thought, well, okay, I do know a little something about yeast. I didn't know very much, but we had certainly heard considerable. And I think genomics is in a similar vein. You know, having had the pleasure to watch the department grow into the Department of Genome Sciences, you know, I vicariously appreciate a lot of what's gone on in the technical things. I don't know how to do any of it. I've never run an Illumina sequencer, et cetera, et cetera. But it's easy to find out what the pitfalls are and what you can and what you can't do, and how hard the bioinformatics can be, and how important that aspect is, and things of that kind. And so in all of those cases, I think I've benefited enormously from hearing what my colleagues and their students were doing and saying and thinking about over time. And it's made a tremendous difference that I can talk to my colleagues at the Sanger Center who are doing the genomics of the malaria parasites and be reasonably knowledgeable about it. And that's been a huge plus, and given me a chance, I think, to do this project that we were just talking about in a way that I couldn't have done otherwise. You know, not that I know very much, but I know enough to ask questions and understand what people are trying to do, even if I don't know how to do it, and I usually don't. But that's made a huge difference to me, and given me a venue, really, that I really wouldn't have had otherwise.
Well, would you have any recommendations for any of these fine people if they're about to hop over to Africa or Southeast Asia? Can you give them a recommendation?
Whoa. Well, I'm not supposed to give you know, clinical advice, and I'm not a clinician. The CDC has a great website. Maybe that's actually, you know, really wear long sleeves indeed. It really is. It's the very best if you're honestly going.
A couple bed nets, would that help?
Bed nets are good. Bed nets are very good. I use a bed net whenever. Bed nets are a pain in the neck, I have to say. I mean, they're hot. They keep you from getting any breeze that there might be. And I do have colleagues who absolutely know better and do not use a bed net. And, you know, figure, okay, make sure you got your treatment dose, because, I mean, you only go to these places when it's malaria season. I mean, you know, you don't go when it's dry. That would be not very useful. So you tend to go, you know, at the height of the malaria season when you know, you've got wall-to-wall children lined up every morning who clearly, most of whom have malaria. So it's not very smart not to use a bed net, but some of them don't, but I do.
So is there time for questions from the group?
Janet.
You were the first woman faculty member in the department. Oh, sorry. Oh.
Ah.
You were the. Is this on? Okay. You were the first woman faculty member in the department. I'd like to ask you how you found that. Did you feel isolated? Just how it was to be the first woman faculty member.
Actually, it was fantastic. I mean, the support from the department throughout this, I mean, genetics and genome sciences as well, has been amazing. And when Tom and I were looking for jobs, we had an opportunity at University of Georgia, and we interviewed and talked with people there, and the contrast between the feeling that you got was very large, shall we say. And in fact, I came from two very large departments where there was considerable competition among the faculty. And I knew I didn't really want that. And this department, to my delight, really did fulfill that. I mean, you know, the various people I've mentioned have been supportive from the beginning. And I don't know, I mean, I suppose, I mean, I'm old enough that I was really used to being the only woman in whatever group. I mean, you all came pretty close to that too. And so that wasn't surprising. But in terms of support for me as a new faculty member, I mean, Herschel, I was talking with Anne earlier, Herschel's daughter, who also was at that stage in her career of beginning a faculty position and starting a family and all kinds of things like that. And I think in both cases, my graduate mentor also was a very gifted man, Gordon Tompkins, and had two daughters as well. And I've often thought that I was a huge beneficiary in both of those cases, that highly accomplished men who have only daughters have to sort of notice that these daughters are the ones who are going to be accomplished in their, you know, in their next generation. They don't have the option, if you like, of putting all their eggs in their, you know, in their son's basket or whatever. And so, you know, I think, you know, well, I'm sorry, this is a bad analogy, but anyway, bad, well, you know, anyway, you get the point. And I'm really sure that I was a beneficiary in both of those cases, that both in Gordon's case and in Herschel, I know in Herschel's case, that there was a recognition that, oh, well, okay, I think perhaps women can do this, because after all, Anne is doing it too. And that was a huge advantage to me, I think. And so, I mean, you know, some of you know Herschel well, and he was one of those people that if you're one of his people, you really were in. And I was lucky enough to be one of his people. So it really didn't cause me difficulty. You know, you hear horror stories from some other places, but not from here, I don't think. Bonnie.
So how many times did you get malaria?
Only twice. I keep asking Raghu to come bring me back some, you know, but.
Well, you seem to be doing your own work.
Well, one of the times I got a rather unusual kind called Plasmodium ovale, and it was really rather ghoulish. I mean, all of these colleagues who wanted samples, you know, calling me and saying, you know, don't get treated right now. You take a little more. You know, I mean, this isn't one that kills you, so it's probably okay. You know, and just take a somewhat longer, you know, a little longer time, a little more blood samples. Send me the blood, and I did a little bit of that, but I mean, you know, there was a limit to what I was willing to be putting up
She
with.
was the main blood source when we were growing the parasites.
This is true.
They really liked her red blood cells.
Yeah, this is also not probably legal at this point, but anyway, we did do that for quite a while. And you never ever let me try you, right?
I may have sickle cell trait
That's
or
right.
something.
Yeah, well,
you
we
know?
could have found that out with no problem, actually. Single base, you know. Anyway.
So, Carol, two questions that are sort of different aspects of the same. Could you talk a little bit, because you've both worked in the lab and in the field, about the tension between lab workers and field workers in malariology? Because this has been cordial sometimes, and I know it's been contentious other times.
Yeah, there certainly is a tension, although the kind of lab things we were doing were already a bit applied. I mean, we were asking about, you know, what kinds of genetic changes were responsible for resistance to antifolates and things like that, which were not completely basic science. The other thing that's happened in the last 5 years, or maybe a bit more, is that the gulf between them has gotten smaller, I think. People who are doing the basic science are beginning to think about trying to translate that into work. And there's, of course, the other big change. There's money for translational things, as you know better than I do, in fact. And so the opportunity to apply, I mean, people, for example, who are very interested in the amazing biology of what the parasite does when it gets into the mosquito, as you know, is really complicated. It goes on, and it's very hard to study, et cetera, et cetera. But if you could come up with some sort of a vaccine that would prevent the transmission through the mosquito, you're home free. And so those folks have gotten much less esoteric these days, and are really trying to think about, well, could I use this to make a useful vaccine? So I think the tension it's not gone, but it's much ameliorated, I would say.
So I guess you partially answered the other aspect of that question. Where do you think, you know, by virtue of having this almost unique perspective on the different aspects of the field and having worked it both in this country and in Africa, where are the big opportunities at this point, from your point of view, to make headway on malaria as a disease problem, not only in Africa, but elsewhere in the world?
I guess, I mean, there's a huge amount that could be done. I mean, at its base, malaria is a disease of poverty when you really come down to it. And so, unfortunately, that, you know, one way to solve the problem in most places would simply be economic development. That's what certainly happened in the south of the U.S., in parts of Latin America. That's certainly what happened in Europe. I mean, Italy was a wall-to-wall malaria for, you know, centuries and centuries. In other places where the transmission has been much higher, it would be a much larger effort. But there's a huge amount that's been done by simple combining what we already know, fed nets, other kinds of preventions and controls, you know, efficient treatment. I mean, prompt treatment with an effective drug is awfully useful. And so putting together all of the things we currently know how to do has already made quite an impact. It'll be very expensive and very difficult to do in some places. I mean, the places where malaria is still rampant are places of civil disorder, as they say, some place like the DRC, the Democratic Republic of Congo, former Zaire. You know, we don't go sample there. I mean, there are places in eastern Congo or in eastern DRC that I would not send anyone that I knew or cared about. I mean, it's a really dangerous place. And places like that, I mean, in Cote d'Ivoire, where we have a lot of colleagues, I had a young man who worked in my lab for several, he's finishing his thesis now in Cote d'Ivoire. And, you know, they've had, again, a huge civil disruption, so malaria goes up again. Pakistan, that had this huge flood, not this year, but the year before, they have huge malaria, huge dengue, another arbovirus carried disease carried by mosquitoes. So those are the challenges. But then you say, well, okay, what would we do about those challenges? You know, these are natural disasters. These are, you know, civil disorder. I don't have any. You know, certainly don't have a simple answer for those. There are lots of places where malaria can be controlled, as it has been here in the United States and in other places I mentioned. So, you know, places that are stable and where the economy, you know, where there's some reasonable economy, malaria can be made not a very big public health disease. Many parts of Southeast Asia. Thailand has done a fantastic job. I mean, they still have all the, you know, the mosquitoes don't go away, but you'd have a hard time catching malaria you couldn't catch it in Bangkok, but you know, most places in Thailand, there's really not much worry. In countries like that, or Iran has done a wonderful job as well. But you know, then Afghanistan is right next to Iran, and they get lots of cases over the border because of what's going on in Afghanistan. So it's a complex political, economic problem in some ways. I mean, there's obviously a disease and lots of things to do about the disease. I mean, you know, whether there will be a vaccine, I don't know. I'm not convinced, but I'd be delighted if it happened. But beyond that, I think we'd have a tough time. But we could do control in so many places and do a really good job at that. And once you get the incidence that you measure by just lining up school children and taking blood samples, if the incidence is below 5%, it's no longer a huge public health problem. It's a manageable problem at that point.
Carol, you once had a result in your drug resistance in which I understood that the resistance to one drug made a weaker resistance for a second drug, and
Yeah.
it seemed to me that was the edge of something really fascinating. And I don't know if that ever turned into anything. Did you ever find a case where that you got a directed insensitivity of some kind?
Yeah. I mean, not only did that work with the two drugs that Jason also worked on, the standard anti-malarial drug, pyrimethamine, and then the experimental one called WR-99210. The WR-99210, and that was the pair where being resistant to one made you sensitive to the other and vice versa. And the WR-99210 is still under development. It's a long story about why it's taking so long. But I think that one may work. And in fact, there's another example of it. Resistance to chloroquine is another very old anti-malarial drug, and the resistance to it is almost worldwide. And it turns out if you carry the alleles that confer resistance to chloroquine, you're much more sensitive to a much more modern one called lumefantrin that Novartis is selling hundreds of millions of doses of in a combination. And one of the jokes is that, in fact, if it weren't for chloroquine resistance, the Novartis drug would never have been developed, because again, the resistant allele of the allele that confers resistance to chloroquine makes you extremely sensitive to lumefantrin. And we're beginning now, one of the things we're doing in this network, we're beginning to see that changing, because now the huge drug pressure is from the lumefantrin, and we're beginning to see the other allele rise somewhat in proportion in the population. Whether you could really play one off against the other, it would have some interesting practical problems, but the idea works, and you can see it in this other instance as well. Evolution really does work, actually.